Hello everyone, myself Akriti Maurya, volunteer of Shiviforensics, and today I am going to present polymerase chain reaction technique. So, introduction. Polymerase chain reaction is a powerful molecular biology technique used to selectively amplify a specific DNA sequence in vitro condition. It denatured the double standard DNA into single standard DNA and then select a segment of DNA to amplify using a specific primer, and then follows the method of replication using DNA polymerase enzyme to amplify that segment. It generates millions of copies of a target DNA region from a very little sample, and it is also known as molecular photocopying. This technique was mainly invented by Carey B. Mullis in mid 1980s, and for that he got the Nobel Prize in Chemistry in year 1993. So next is ingredients. So mainly these are the ingredients which we use for the PCR techniques. Like first is DNA templates, and the templates which we have to amplify will take that DNA templates, and then primer. Primer is basically artificially synthesized 15 to 25 nucleotide in length. It should be shorter. The shorter the primer, the better the elongation will happen. And then third is tag polymerase, which is thermostable and it is extracted from Thermus aquaticus bacteria. Then we also use the free magnesium ion, which act as a cofactor for DNA polymerase activity. Then fifth is DNTPs, which is deoxynucleotide phosphates, mainly adenine, guanine, thymine, and cytosine, which act as a building block of new DNA strands. Then all these process completed in a tris HCl buffer, which pH ranges between 8 to 9.5 depending upon the medium. And then fluorescent dye. Fluorescent dye is mainly used in quantitative PCR or digital PCR. Otherwise, in real-time PCR, it is not required. This is the PCR machine. This is how it looks like. Mainly, we take all the ingredients into a PCR tube or microplates, and then put it in a thermal block, and then we close the lid, and everything the time, temperature, and cycle, everything present or shows in on display, and according to that we use this machine. Then there are mainly three steps involved in this whole cycle. The first is denaturing stage, on which the temperature is very high, and so that double standard DNA can separate into two single standard DNA. And then annealing stage, in which temperature is lowered, and then temperature in again increased, and which is also known as extending stage. So the first is denaturation stage. It is last between 5 to 10 minutes at 95 degrees centigrade. Mainly reaction temperature increased to 95 degrees centigrade, which melts the hydrogen bond between the DNA, splitting into two single standard DNA. Then second step is annealing. It mainly lasts between 30 seconds to 1 minute at 45 to 60 degrees centigrade, and the temperature is lowered to approximately 5 degrees centigrade below the melting temperature of primer, to promote primer binding to the template. Melting temperature of primer, we can calculate by this formula, which is 4 bracket में G plus C plus 2 A plus T in brackets. And then comes the next stage, which is extension. Mainly this in this process, like in this step, we increase the temperature again, so that extension or DNA polymerase function properly and extension happen. So mainly the temperature is increased to 72 degrees centigrade, which is optimum for DNA polymerase activity to allow the hybridized primer to be extended. Then this process is repeated up to 30 plus cycle, uh, involving in all the steps, resulting exponential amplification of a specific DNA sequence. Then this is the whole process, like shown in the diagram. First we take the DNA, and we and the temperature is increased, so that denaturation happen, and double standard DNA is split into two single standard DNA. And then here primer came and bind with its specific complementary sites of DNA. And then polymerase came and bind, and DNTPs elongates the process. So DNA polymerase mainly elongate the primer by sequentially adding the nucleotide and generate double standard DNA by following the process of DNA replication. And this whole process, like denaturation, annealing, and extension, is repeated up to 30 plus cycle, so that we can get the number of specific like DNA sites, and we can use further in our investigation process or anything else, like whatever the sequence we have to find. Then here is the application, like it mainly used as infectious disease diagnosis. Like recently uh, in COVID-19, there are lots of uses of PCR. We have seen it, like mainly RNA to mainly we used RT-PCR for COVID-19. Then detection of gene mutation or genetic disorder. Also DNA fingerprinting, like in disputed paternity or maternity cases, this PCR works really well. Like even from a very small sample, we can get multiple of specific sites or multiple of copies of our DNA, so that it can easily determine, or we can easily match in disputed paternity or maternity cases. Then it's also used in DNA or RNA quantification. Like for RNA, it mainly converted into first it converted into DNA, which is also known as complementary DNA, like through the RT-PCR process. And then it is also used as a genetic uh, as a tool in genetic engineering. These are the references which I use to make these slides, and thank you.